So that being said, looking for the good Mexican virus for today for Cinco de Mayo, but unfortunately, unable to find one. Although the swine flu that we talk about towards the end is close to that. So at least we have a little bit of the Mexican approach today. But of course, first we have to have our clicker questions. The structure of filovirus virions is most similar to blank virions, poliovirus, flavivirus, paramyxovirus, rhabdovirus, or everybody's favorite bacteriophage lambda. Nobody liked my bacteriophage lambda. How could they? But no, filoviruses look nothing like lambda. So at least say the virions thereof don't. What kind of virions do polioviruses have? I know it's a couple weeks to the midterm, right? So we're not too worried about it yet. It's our naked icosahedral pseudo T equals 3 icosahedra. Well, flaviviruses. They're enveloped, but otherwise very similar to poliovirus with a really flat structure because the envelope proteins sit down on the outside. Paramyxoviruses, they're enveloped, kind of blob-like, rather like actually what we're going to be talking about today. The rhabdoviruses, kind of bullet-shaped, and bacteriophage lambda, of course, head and tail. So they are much more like rhabdoviruses and paramyxoviruses. One could probably argue about this, but since I had in the lecture notes they're much more like the rhabdoviruses, we'll use that one as the correct answer. Anyone happy with that? Or everyone except for the person who voted for this one? Okay, next question is what kind of protein is packaged in filovirus virions but not in rhabdoviruses? RDRP, membrane glycoprotein, matrix protein, nucleocapsid protein, transcriptional activator. Dee 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 doo dee dee doo. 3, 2, 1, lift off. So RDRP is what? Yes, thank you. RNA-dependent RNA polymerase. Membrane glycoprotein. Yes, we've definitely got them in filoviruses. What about rhabdoviruses? What's it called in rhabdoviruses? At least in say vesicular stomatitis virus. It's the G protein. So G should be a really good indication that it's a glycoprotein. Matrix protein. That's what makes the rhabdovirus form. Nucleocapsid protein. All negative strand RNA viruses. So we're left with transcriptional activator, even if you didn't remember what the name of it is, which is PP30. Yes, exactly. Last question for today. The likely host/reservoir species for Ebola virus is humans, chimpanzees, bats, civet cats, or antelope. Pardon? They're always trick questions when I ask them, right? By definition. Sure, yeah. It probably has to do with this up here, I'm guessing. You know, host versus reservoir species, exactly. Oops. So, what was your question? So yeah, so the question really has to do with the main point of this question, which is what is a host/reservoir species? And what is a particularly reservoir species, but also probably the natural host? I didn't want to put natural host in here because that's not really the standard terminology. Reservoir is much more of the standard terminology. What's the definition of that? I'm hearing mutterings here. So not necessarily no sign of disease. And so that's what a lot of people may get confused about. It's where it's being maintained in a population. Usually that's not having any particular sign of disease, because it would make sense that it be maintained in that part of the population. But there are plenty of reservoir species that do show disease phenotypes. It's maintained in that particular population. Whereas Ebola is clearly not maintained in humans because they all die. Well, most of them anyway. And chimpanzees is very similar. Bats don't seem to cause much kind of phenotype. We can't tell if they're really sick. And a lot of them have the virus that's associated with them. And so that's really how you think about these things. And we actually don't know, and that's again why we've got likely host. We don't necessarily know. We've been screening bats, but that's because they're sort of a usual suspect kind of thing. And so it may or may not be that these really are the hosts and reservoirs for Ebola virus. Civet cats were thought to be what? Host? SARS, exactly. So they're thought to be hosts for SARS. What about antelope? Now, I was never quite sure. Is antelopes the appropriate plural or antelope the appropriate plural? Antelope is plural? Like deer. Like deer, antelope. Deerzes. So, but why? Why did I put antelope on here? Yeah, because the antelope can be infected, and often they have a really also nasty, exactly the same kind of phenotype as you have with humans and chimpanzees. And they're a nice indicator species as opposed to a reservoir species in terms of looking at that. But yes, it's probably the bats, but we're not completely sure because we haven't screened everything which is out there. And so that's the current theory as far as that's concerned. So as far as the filoviruses are concerned, so here's our cute little Ebola virus that was taken in a BSL-4 lab because it's the only place you can work with these things. The genome, again, is very similar to the paramyxo and rhabdoviruses with the slight differences of the overlapping start and stop codons, which is a pretty minor difference. Proteins, the main thing, of course, is the VP30 transcriptional regulator. Editing, again, very similar to paramyxo and rhabdos, but here it's having to do with the membrane binding or not binding of that glycoprotein. Regulation, again, the only difference here is really that transcriptional regulator, which is involved in the RNA structures, and then, of course, these nasty hemorrhagic fever diseases, and I still haven't changed that particular aspect. There are more questions about the filoviruses before we move on. One of the days that I'm not going to be here, we'll have a video that talks a little bit more about Marburg fever. So you can get to see some really nice real-life examples of Marburg fever. How nice. So on the other hand, we're going to talk today about viruses that kill orders of magnitude more people, but don't get anywhere near as much press. So the orthomyxoviruses as opposed to the paramyxoviruses, these are the real myxoviruses. Where that came from, no idea. But the important thing, certainly at a molecular level, is that these are now segmented. So it's not just one single RNA, which is the virus genome. It's multiple small RNAs with the virus genome. And I really like to think of these like chromosomes, like a viral kind of chromosome. And so that's the segmented aspect of thing. Of course, most people think about these in terms of influenza, and then I'll do a diatribe on vaccination towards the end today. Yet another diatribe maybe is better. So key concepts about these true myxoviruses. Again, it's a segmented genome. We've talked a little bit about hemagglutinin and neuraminidase already, but again, this is the classic system where we understand the most about the receptor binding proteins on the outside of these viruses, and then some of the activities that we have also, and this is in the case of influenza, is a separate protein, the neuraminidase. H and N, you'll be hearing lots about if you haven't heard already. H1N1, for instance, is hemagglutinin 1, neuraminidase 1, and so on and so forth. So also talk a little bit about the matrix and ion channel proteins. One of the matrix proteins is, in fact, an ion channel as well. And that has to do with the conformational change that you have with the hemagglutinin protein due to changes in pH. What's really different we haven't talked about so far is nuclear replication for these RNA viruses, which is really kind of bizarre. Why would you want to be replicating in the nucleus if you're an RNA virus? We'll see why that is, and it has to do with the fact that that's where the caps are. And that's why you want to do that. So cap snatching. So the orthomyxoviruses are well known for going into the nucleus, again, totally over-anthropomorphizing here, and stealing the caps from messenger RNAs that are already made. And this is a really, again, smart thing to do because that way you don't need to make your own caps. They're already there. And so you can just use those, again, for translation. Of course, that has the same problem that you've got to get into the nucleus, because that's where most of these capped messenger RNAs happen to be. So that's how you go where the caps are. So origins, again, we'll talk about disease at the end. Structure, the structure of these guys is really kind of blobby again, rather like what you see for the paramyxoviruses. I love this micrograph because you can see here the individual chromosomes in this particular virion, which is really to me, kind of mind-blowing. You can see those individual ones in here. Again, binding and entry, it's all about hemagglutinin, changes in pHs. Messenger RNA production and replication, again, these are the things that we haven't really talked about so much already. Getting out is pretty straightforward, and then disease is all about this guy. Influenza, there are 3 main kinds of influenza, A, B, and C. A and B are well, say A is probably, I'm trying to remember the numbers here, about 90% of disease, B 19, and C maybe 1%, so much, much lower. It's not what's caused by rhinovirus or coronavirus. Everyone says they have the flu. Well, 90% of the time they don't have the flu because they wouldn't be telling you that they have the flu. They'll be home in bed. So we'll be talking much more about this as we go on, again, particularly towards the end of today's lecture. Orthomyxoviruses, they're, again, enveloped viruses. They bud at the plasma membrane. Here's a, again, relatively famous picture of influenza. Blobs, yes, but also these long filaments that form. And so it seems to be multiple different forms that you can get of these enveloped viruses. Doesn't seem to make much of a difference. You know, it can be a blob, can be a filament, lots of different things. So the matrix doesn't seem to be that critical in terms of how the virus particle is put together. So really quite like the paramyxoviruses as well. But again at the plasma membrane, so this is a cell where the virions are budding off of it. Packages 6 to 8 nucleocapsids, and those are those individual chromosomes. And this is really the big difference between the A, B, and C forms of influenza, just the number of different chromosomes that they have. And again, sometimes they're forming as these filamentous forms as we have over here. It's a lot easier to think about this, at least for me, in a cartoon form. Of course, it's not really what it looks like. But this is a typical Influenza A virion with 8 of these nucleocapsids here that are bound to nucleocapsid protein, which is actually not shown on this slide, but each of these supercoiled mini chromosomes is coded by the nucleocapsid protein. Associated with each one of these is the replicase complex, which, as we'll see a little bit later, is composed of 3 different proteins. Again, RNA-dependent RNA polymerase. But each of the chromosomes has one of those associated with it. So it's actually not unlike what we talked about for filoviruses and the paramyxoviruses already. The replicase is already associated with these guys when it comes inside the cell. But again, they're negative strands. They've got to, first thing they need to do is replicate their genome in order to be making messenger RNA. So that's what's present on the inside. There's also these non-structural proteins, NS2 and NS1. We'll talk a little bit more about those later. Probably mostly involved in pathogenesis. And who cares about pathogenesis? No. But mostly involved in pathogenesis aspects of things. Then as you move out, you've got another matrix protein. Turns out there are 2 different matrix proteins. M for matrix protein, M1 and M2. M2 is probably the most important as far as we're concerned, again, because it's different in these viruses than any of the other ones. And it's an ion channel. Is the target of some drugs that unfortunately don't work very well anymore. But that's the ion channel. Then, of course, our friends, the hemagglutinin and neuraminidase proteins. These are what's sticking out on the outside of the virus particle. Hemagglutinin is binding to the receptor. The neuraminidase seems to be involved in chopping off sialic acid on an already infected cell so that the virion can be released. So these are the genomes, so the genes which are packaged, say the gene products that are packaged in here. You encode 10 proteins in the genome. 9 of them are actually packaged in any individual virion. So basically every single protein which is being made ends up being packaged, even those that people call non-structural. So it's unfortunately a little bit of a confusing nomenclature here. Wanted to mention a few of these. Again, this is just that list of the different proteins, those non-structural 1 and 2 down here. Hemagglutinin, we've talked about a lot. Again, it's that surface protein that binds to the sialic acid, then has a big conformational change on changes in pH. How does that change in pH take place? It takes place in the endosome, but you also have a pH change that happens inside the virion, and that happens through the M2 protein down here. So it's a conformational change that happens in nucleocapsid protein as opposed to what's happening in the HA protein. We'll look at that as we look at entry a little bit later on. Then we'll also talk a little bit more about PB2 and PB1. These are parts of the RNA-dependent RNA polymerase. Most important is PB2, because that's the cap stealing aspect of these viruses. Turns out that a lot of the attenuated vaccines, strangely enough, have mutations in these polymerase proteins, and we'll get back to that a little bit later on. But first, I want to talk more about HA. This is the structure that we've seen already. This is just one monomer of the trimeric structure over here on the right-hand side. It's made originally as one polypeptide, then gets cleaved through cellular proteolysis. There's no protease encoded anywhere in the influenza virus genome. So it's a cellular protease which cleaves this protein. There's a cartoon over here. Starts out as being one whole protein, gets cleaved, and that cleavage generates the fusion peptide, which is normally hidden inside the structure until, of course, you have your conformational change, which happens. So if we look at that conformational change, again, we've looked at this quite a lot of detail already. Here you have binding of HA to the sialic acid, receptor-mediated endocytosis. In the endosome, the pH changes. That pH change leads to changes in the structure of the HA protein. So up at the top here, first you have binding, fusion peptide, which is this little red thing here, hidden down at the bottom of your protein. As soon as you have the pH change, these guys have membrane fusion. Then you have the fusion of the two membranes, and it's the last step right here where you get release of your nucleocapsid proteins. That's dependent on the change of pH inside the virion. And that comes through the M2 protein, which is blocked by these antiviral drugs. So it's an antiviral drug blocking uptake. So it's that entry system which is being blocked. Unfortunately, since it's been used so much and we've been exposed to a lot of these influenza viruses for many, many years, 90-some% of circulating influenza viruses are resistant to this particular drug, or even this class of drugs right now. Let's look again at the influenza HA protein. Starts out over here on the left-hand side as HA0. That's before you have any proteolysis that takes place. The proteolysis is right here in these yellow loops down at the bottom. Cleavage in this yellow loop then gives you the two proteins, HA1 and HA2. The fusion peptide is buried down here until you have the conformational change, which happens at low pH, which gives you these longer alpha helices and again sticks the hydrophobic helix into the other membrane. So again, we've talked about this before, but just wanted to remind you again. So once you have this, once you have the change in the pH inside the virion, then you release the nucleocapsids. And so these are the cartoon form down here at the bottom of these nucleocapsids. Here you can see the NP protein. Again, usually when N is going to stand for nucleo something or other. So we've had N proteins, NP proteins, NC proteins. These are what's binding to these genomes. And so these are electron micrographs of what these structures in fact look like. So if you purify these structures from influenza-infected cells, you get these circles with blobs attached to them. That's about the highest resolution as we've gotten so far. But you can, by looking at where each of these individual proteins bind, you can get a pretty good idea of where each of them fit in the structure. So probably you don't have this nice helix down here. It's probably much more of a larger circle where you have binding of these nucleocapsid proteins. It seems very similar to the case with paramyxo and rhabdoviruses. Those nucleocapsid proteins stay bound most of the time. So it's the RNA is on the outside, so it actually can be used then as a template. So usually binding with these things. What is using that template? It's the polymerase proteins, which are all bound at the 5' and 3' ends. And so the 5' and 3' ends we know are very close to each other in each of these subgenomic segments. Again, I like to think of them as chromosomes. where you have binding of all three of these polymerase proteins at that point. So this complex, which is in fact pretty big, if you think about it, I think, if I remember correctly, this is 100 nanometers, the scale bar on here. This whole thing has to go into the nucleus. And so that process of getting each of these individual nucleocapsids into the nucleus is, again, extremely specific for these true myxoviruses. So what happens, again, you have all the conformational changes, the change in pH, each of these nucleocapsids, remember there's 8 of these in any Influenza A virus. These guys have to get imported into the nucleus. First thing that happens is that these polymerase proteins also steal caps and now make messenger RNA. That messenger RNA is exported back out into the cytoplasm where it gets translated because all the cellular translation is happening back in the cytoplasm. Then each of those proteins gets re-imported back into the nucleus. They've all got nuclear localization signals to assemble new particles, which are now these individual nucleocapsid segments, which then again have to get exported. So if you think about it, this is really convoluted process. Now, if you are going to go where the caps are, you've got to go through all of these molecular gyrations to get yourself in and out and all the rest of it. So it's, makes sense from the point of view of getting the caps, but it means you've got to do a lot of nuclear import and export. Now, fortunately, for the virus's point of view, that's not something you can really treat disease-wise, because nuclear import and export is something that clearly you need for all of your cells pretty much all the time. So it's, again, unfortunate from a treatment perspective, but is a rather complicated system in terms of moving all of these things around. One thing that you probably notice on here is that there's no HA or neuraminidase shown on here. That's because these, of course, are being expressed in the endoplasmic reticulum and going to have to go out to the membrane. Because these are membrane proteins. These are all budding at the membrane. But to get this process, how do you go about getting your messenger RNA in the first place? As I mentioned already, it's all about cap snatching. So the PB1 and PB2 proteins will bind to the cap, normal messenger RNA, present inside the nucleus, and cleave off that particular cap structure. And it always cleaves at an A residue because the 3' residue of each and every single one of the Orthomyxovirus nucleocapsids is U. So binds to, cleaves that off. Then you've got their viral genome RNA. And remember, this is all mixed up together. It's got the nucleocapsid protein, all of them bound together. Then we'll make a copy of your negative strand over to a few U residues at the end of the genome, right before you get to this opposite end, which base pairs back over here, because the 3' and 5' ends are right next to each other. Get to this set of U residues, just like you have with paramyxos, phylos, rhabdos, etc. You have stuttering that takes place, and that generates your poly A tail. So these RDRPs, particularly PB1, is really good at making a whole bunch of A residues. So you have that down here at the bottom. So steal the caps, stuttering. There are a couple of cases, in fact 2 genes, 8 chromosomes in Influenza A, 10 proteins that are made. There are 2 which are also spliced, and so there's some splicing which takes place as well. Yeah?
So when you say that each of the nucleocapsids starts with a U,
Mm-hmm.
each of the nucleocapsids, is that a complete genome or just like part of one?
So each of these will be your segment. Each of the, in the case of Influenza A, 8 segments, starts with U, and in fact UC, at the 3' end, because remember this is a negative strand. At the 5' end, it's, well, again, reading from 3' to 5', really should be reading the other way. But here, GA going out to the 5' end. So the 5' end is an A, which can then base pair with the U. And then the second base, again, going from 5' to 3' is a G, which can then base pair with C. And that's true for every one of the 8 segments. What's present in between here, which is where your messenger RNA is going to be different for each of those different segments.
So if there's
Yeah, so if one of those genome segments, for instance, would have mutations at either of your terminal nucleotides at the 5' end or at the 3' end, that's not going to be functional anymore. Yeah, so that would be a non-functional mutation that takes place. So this is how you get your messenger RNAs. That's all well and good, but of course you also need to make genome, right? So how do you make genome? Again, it's very similar to what happens in Paramyxos, rhabdos, and phylos. It's all dependent on the amount of your nucleocapsid protein. So if you've got large amounts of nucleocapsid protein around, and this is nucleocapsid protein that's not bound to genome, because you remember when it comes in, it's all bound to nucleocapsid protein. If you have free NP protein, that binds to the RDRP and basically says, hey, stop snatching and don't stutter anymore. And in that process, you end up just making first anti-genome sequence, because you've got negative strand, then you make the positive strand, and then that positive strand gives you template to make your negative strand. How you get more of your negative strand than the positive strand is not entirely clear. Actually, in Orthomyxovirus infected cells, you have quite a lot of positive strand, probably much more so than you have in some of these other viruses as well. So we've made our messenger RNAs, we've made our genomes. What about these other proteins, the NS2 and NS1? NS2 is a nuclear export factor, which makes perfect sense. Once you have assembled your whole genome, you need to get them out. NS1 is really kind of interesting because it blocks the classic tailing proteins that you have for regular messenger RNA development. So the poly-A binding protein and the cleavage and polyadenylation specificity factor is what you need for normal 3' end formation in cellular messenger RNAs. Why would it be useful to block this for the virus? What happens to normal messenger RNAs, these are now cellular messenger RNAs? They get cleaved and poly-A tailed. But why does that have to happen? Right, they're not going to form the circle, but what happens once you've actually formed the circle and made sure that you've done all your splicing and poly-A tails? What happens then? After that, your messenger RNA gets exported from the nucleus. If you haven't made a proper poly-A tail, then the RNA doesn't get exported from the nucleus. If the RNA doesn't get exported from the nucleus, what do you have? A whole bunch of capped messenger RNAs that haven't been exported. So if you block tailing, you end up with more caps in the nucleus. So that makes perfect sense. Blocking protein kinase R, way back when we talked about entry and antiviral defenses, PKR is protein kinase stimulated by double-stranded RNA, which normally shuts down translation, which would be a really good thing if you're worried about translating a whole bunch of viral proteins. But NS1 blocks that activity. And so you can still get lots of that. Seems to happen mostly through basically pretending that it's double-stranded RNA, and so blocking that. If you remember, NS2 was actually present in the virion. NS1 is not present in the virion. This is one of the only proteins that doesn't get packaged in terms of the virion because you only really need it much later on in the whole process. So once you've put all these things together, now you need to package them. Hemagglutinin and neuraminidase have already been made in the endoplasmic reticulum. Those get transported out to the plasma membrane. Then you get your RNP. What's an RNP? A ribonucleoprotein complex. So it's just each of your individual genome segments associated with nucleocapsid protein. That then gets packaged into each of these virions, which end up getting released. What is probably one of the major questions that we really don't have a good answer for in terms of influenza virus biology is how the heck do you get just 8 of these things, and how are they specific that it's one and only one of each of those individual segments? That nobody has a good idea how that's actually happening, because it's hard to see where you'd be getting the specificity from. So this is, again, a really open question in terms of how all of this is functioning. I wanted to show this slide. This is from the other textbook that we're not currently using, but I think this is a really nice overview of that whole process. So just a quick review. First thing that happens, number one, up there on the top, binding again to HA cellular receptor, which is sialic acid. Endosome formation, release of the genome, which happens at 2 there. These subgenomic segments, number 3, gets imported into the nucleus. Then there are 2 processes. First, you make all of your capped messenger RNAs. That's through 4 here. Some of those get exported, of course, translated. The proteins come back in. And then some of these cases, you're making proteins which are going to end up in the endoplasmic reticulum, which get targeted down here to the membrane at the bottom. Once you've made these proteins, we haven't talked about NEP. We can ignore it for the time being. It's one of those extra splice products. But NS1 come back into the nucleus. Again, that's blocking what you have in terms of tailing processes. So you end up with more of the host messenger RNA. Once you get enough of your nucleocapsid protein, you shift over to making genomes. Those genomes get packaged, and then magic occurs, and you get the assembly of your 8 proteins which are left and leave the cell. Any questions about how these guys do their thing? Now we can shift over and talk about what they do to us when we get infected. So I apologize for all the text on this. I wanted to be correct, and so I've downloaded a whole bunch of stuff from the CDC as far as is concerned. Basically, your flu symptoms are basically kind of whatever you get with a cold, except they're nasty, really strong. So, and again, the quote from our textbook, if you can't get out of bed, it's probably influenza. But if you can, it's probably not. A lot of people talk about stomach flu. You can have some gastrointestinal effects, very rare. It's really a respiratory disease. So people talk about stomach flu. It's not the flu, or very, very unlikely that it is the flu. So what's happened this year? Great details on all the things that are going on. This is literally from, I think, 2 weeks ago, if I remember correctly. Yeah, so it's the 18th week. So this was in April. What this flu season was like. Just looking at here, number of positive specimens. This is definitely an underrepresentation of the number of influenza cases, on the order of about 4,500. Peaked right at the beginning of the year. So just down there, this is just number of weeks. So 2013, week 52. 2014, week 02. So really right around New Year's. This is pretty typical in terms of influenza. If you look at the different kinds of influenza we had, most of them, the orange bars here, are 2009 H1N1. That's the swine flu from 2009. And so that's the major form which is circulating right now and causing disease, again, because these are the people who are coming in. Curiously enough, we have a bit of a bump now, which is influenza B. Usually causes much less severe disease, but often we'll have this kind of bump a little bit later on. So this is what this year looks like. A relatively normal year, and the way we know that is just by comparing to previous years. So this is 2013, 2014, right here. Here's our normal range in any given year. The black lines here, the red lines are the percentage of deaths in any particular year due to these infections. And this year was actually pretty bad, relatively speaking, in terms of percentage of diseases due to these kinds of infections, which probably has to do with the fact that it's H1N1, the 2009 version, which seems to cause a little bit higher mortality than any of these other ones. Again, this is week 26, so just a couple of weeks ago now. But again, we're coming back down. Very seasonal, what you see here. In terms of actual numbers of deaths, these are pediatric deaths. varies from year to year, whether you have more deaths at very young or very old patients. But it's not too different from other years. 91 this year so far. We're coming down. Last year there were considerably more. 2011, 2012 was pretty small. 2010, 2011. So on the order of 100 or so pediatric deaths that you get any individual year from flu. Now let's just step back a little bit. Think about total number of people in the world. This is in the U.S. that have died from Ebola or from SARS or from MERS, etc. And where is all the media? It's on all of those things. And this is every year. And this is hundreds of kids, let alone the others that are involved. If you look at hospitalizations, which is probably a little better way of looking at this, you see that you have different age groups here. The top line is 65 plus, and in most years that's where you have most of the people coming into hospitals for influenza diseases. But the third line here is 0 to 4, and so this is the young children who are getting brought in. Here, much lower levels that you have, and this is literally from a couple of weeks ago now. If we look at the age groups again, this is a breakdown. 65 plus, 50 to 65, about 50%, fewer here. This year, we have a relatively small number of 0 to 4. It's been going up, going down from 2009, 2010. This was when we had the swine flu. Major thing to notice here is the 18 to 49s, which were really very high at that point, and also quite high for the 0 to 4-year-olds. More data, ridiculous amount here. Don't bother to look at it, but if you think about, people talk about deaths from influenza. Well, almost all of the cases, a death from influenza has some kind of underlying condition, and that's just these underlying conditions which are on the side here. Asthma, particularly for children, up at the top, cardiovascular disease, etc. And then this condition here, pregnancy. I'm not sure I would call that a condition per se. But I would definitely, and never having been pregnant, never likely to be pregnant, but if anyone in my family were, I would like them to be considered for hospitalization if they're coming down with these nasty symptoms. So again, talk about disease and or hospitalizations. Usually there's something else which is going on as well. And so you talk about the number of deaths from influenza. It's probably more deaths than you would otherwise have due to influenza versus anything else. Just look at the number of visits. Again, I mentioned this is a pretty normal year. Right around the beginning of the calendar year is when you have most of your spikes, but this can vary from year to year. So if you look at, this is 2009, 2010, it was very, very early. So this was October, November, where you're getting the big peak of hospitalizations, as opposed to, again, a normal year. Sometimes it's late. So here we are out in week 8, which is what? That's end of February, beginning of March. Some years, 2007, 2008, almost all the hospitalizations were there. So it's not too late to get vaccinated, is the basic take-home message here. There are still some places in the U.S. which are reporting that they have influenza activity, mostly here in the Four Corners area and then in the Northeast. That's probably because the weather's been pretty nasty out there this year. And why there's this correlation between nasty weather and disease is a really interesting question that we don't have a great handle on, but it's probably just mostly people being next to each other. Yeah?
So this is characterizing the type of epidemics that are being observed.
Yeah, well, it's basically the amount. The amount of, and these are certain numbers that you see in terms of relative amounts. So if you were to have looked at a map like this, and I probably could have brought up week 1, basically the whole country would have been brown. So it's, this gives you a really nice breakdown of where things are. So this is, again, the whole country. What about here? What about Oregon? Well, this is what Oregon looks like. This was, again, through a couple of weeks ago, 26th of April. Main peak right here. Mostly older people were hospitalized. But interestingly enough, there were, in fact, more hospitalizations this year than even in 2009. So it was a pretty big year. And we looked at that before. It's been pretty major. And we can look at that relative to different years. This is this year. Here's 2009. It's a little hard to see here, but you should see there's a lot more red down here in 2009 and 2010. That's for the under 5s. And a lot more of the blues over here in 2013 and 2014. It's a lot more of the older people. And so that's the big difference that you see between those two. But in Oregon, this is a pretty crummy year as far as influenza was concerned. So if you average all this stuff across far too many years, think about any given year. And this is very hard to tell because it's only the people who are really sick and usually underlying conditions that are ending up going to the hospitals. Those are the numbers that the CDC has. So you have to do a lot of back-of-the-envelope calculations, and people come up with a 5 to 20% infection rate in any given year. So you do the back-of-the-envelope math, 300 million people in the US, 15 and 60 million people in any given year are infected by influenza, which is a pretty ridiculously large number. And also just think about this class, about 60 people in it. 20, but of course all of you have been vaccinated because you're in my class, right? So 200,000 hospital visits, around 30,000 deaths per year in the US, again, additional above and beyond these complicating conditions. And so again, just compare this to number of SARS fatalities ever, Ebola, etc. So this is completely swamps any of the rest of those. Most of those deaths are people over 65. There are around 500,000 to 1 million every year that die from influenza diseases. Yeah?
Is there any data here on, in Oregon on the pickup of vaccination? In other words, how many people actually got vaccinated for the flu here in Oregon as opposed to the rest of the country?
So there are data for that. I didn't include them in here. One of the really nice things about influenza is that there are ridiculous amounts of data which are out there. And so this, let's go back real quick to this. I don't have the reference here, but I'll make sure I add it. So there's, it's not flu view, that's the whole United States. It's the flu bites, I think. I think it's flu bites, if I remember correctly, for Oregon. So you can, I think you can get that data. I don't have it with me. Putting this together last night got longer and longer and later in terms of putting it together. So again, 30-some thousand in a pretty well-developed world and 500,000 to 1 million worldwide a year. Yeah?
Okay. So that last statistic is deaths worldwide.
Yes, that's worldwide deaths, exactly. So very, very large numbers here. One thing I did want to mention, of course, was what we didn't have on that previous one, and that was our friend in 2009, 2010. You remember for pediatric deaths, and this is under 5, usually on the order of about 100 or so every year. But in 2009, 2010, we had 250 plus. So very large numbers of people coming down with the swine flu. So swine flu really wasn't swine flu. Quadruple reassortment. So the reassortment gets back to that thing I was talking about, packaging. You remember 8 and only 8 get packaged per virion? Well, if you have multiple infections that takes place at the same time in any particular organism, each of those in any given cell, you've got those multiple genomes that are there. Those then, depending on how they get packaged, as long as you've got 8 and only 8, that's going to give you a new virion. The problem is the immune system of whatever organism that then goes into. And so you can have these reassortments that take place, And basically, it's just mixing and matching each of the individual genome sequences. So what had happened in our swine flu, it was mostly human. So some humans are pigs, probably. But yeah, mostly human, but 2 swine segments and 1 avian segment. And it happened really early in the season. And so you saw that bump really happening mostly in October, November, as opposed to later on. A lot of people hadn't been immunized at that point. So that was, I think, one of it. Was really tough on people who are under 65. So very different in terms of a lot of these other aspects. 87% of the deaths. Neuraminidase inhibitors were still working at that point. We'll talk about neuraminidase inhibitors a little bit more a little bit later on. Pandemic was over in August of 2010. So basically came up, went down again. So that was a problem. As we see now, it's mostly what's circulating now is this particular kind of mostly human and partly swine in terms of the reassortment. How do you get the flu? Well, anybody who doesn't have a free seat around them is in exposure range. Coughing and sneezing, again, about 1 meter away. Big problem with flu is you can infect somebody else before you get symptoms and up to 7 days after you've had symptoms. And so that's where you get the big spread, is that you don't really have any of these nasty symptoms, can't get out of bed until it's had a chance to spread, or if somebody comes back too soon. And so that's also a really major problem. What do you do if you get the flu? Well, you can take antiviral medications. There's far too much on this slide for me to read, but basically, antiviral medications for influenza don't do much. And are only really appropriate in people who are being hospitalized or have some other underlying condition. Because basically, you've got to take a lot of it. It doesn't do much for you. Main thing, chicken soup, bed. That's about it in terms of what you can do. And again, also stay home. What are these antiviral drugs? I mentioned the M2 blockers. Unfortunately, almost all of the circulating influenza right now is resistant to all of these. And these are just mutations that have happened in that M2 protein that these drugs no longer bind to. On the other hand, these neuraminidase inhibitors, which again are allowing the viruses to release from one cell and then potentially to move to another. Tamiflu in particular gives you 1 day less of symptoms if you take it really early. Again, not terribly useful. But of course, what you really need to do is go to KFC and get your Tamiflubecue for dipping your stuff into. Now, what you really should do is get vaccinated. And so these are just a few things about vaccines. First started in 1945, so we've had these vaccines for really quite a long time. There are 2 different kinds of vaccine, either attenuated, we talked about before, passaging through cell culture multiple different times, or killed. I love killed because it means they have to be alive, right? Or inactivated in some ways is your classic flu shot. These have 3 different kinds of influenza in them, usually an H1N1 type, and again, that's a hemagglutinin 1 and an N for neuraminidase. And that's each of those segments. They've named each and every single one of them. I think we're up to about 14 different hemagglutinins and something like 19 different neuraminases. 1H3N2 and 1 B strain. This is what they used to have 2 years ago. They introduced a second B strain into the vaccine. So now there's 4 different viruses which are in there. What we had this year was the H1N1-like from 2009. 99.8% of the 2013-2014 isolates matched this. So it's a really good year for the vaccine. The H3N2, 97%. So again, really good choices here. For the B strains, which cause usually a lot less disease, the trivalent vaccine had 72%, but if you included the tetravalent vaccine, it was 100%. So for next year, it's going to be exactly the same thing. And that's interesting because usually every year they change the influenza vaccine because they do this whole recombination business. That you've got the different segments that recombine, or you have genetic drift that happens. These RDRPs don't have any kind of proofreading activity, so they end up putting in a lot of mutations. So you have differences every year. Now, what's nice or not so nice about this is if you notice what I have here, this is for the northern hemisphere. That's whatever we're having here. Well, I also saw those peaks that were happening every year. That happens around January 1st. Well, that, of course, is the opposite season in the southern hemisphere. So you actually have a low point in the southern hemisphere around January 1st, because it's midsummer, and a high point in midsummer. So what happens is the northern hemisphere helps out the southern hemisphere by saying, hey, these are the things that you probably want to be using for your vaccine, and then vice versa coming around. Because it takes about 6 months for any of these vaccines to actually be made. Because it's every year, and the whole process really takes a rather long time. Why should we vaccinate? No symptoms in 70 to 90% of the time. So vaccination is not 100%, and you can certainly be vaccinated and still come down with disease, but works generally much better, less hospitalization, less mortality. If you believe what the vaccine manufacturers say, every dollar spent on vaccines, $30 to $60 is saved in hospital costs, and insurance companies should darn well better be paying for vaccines, because they should be saving over $100 per vaccine that they pay for. So not all of them do, but that's a different story. So I just wanted to spend last couple of minutes talking about my favorite new influenza vaccine. This is the LAIV, so live attenuated flu. This is, unfortunately got chopped off on the side here. This is this recombination, which can be a real problem. Again, if you think about the swine flu or the partially swine flu, 25% swine. Here, basically, what's done is you do this reassortment on purpose. And that process where you take here these two viruses, you infect one particular cell with it, and then you get a reassortment of in this case, a cold-adapted virus with the wild type, which is the normally adapted virus. And then you go through and pick and choose which of these you actually want to use. So in the case of the flu mist, you have 6 segments which are this cold-adapted, and 2 which are your wild type. I can't remember which ones they are. But the idea here is that what you have is now a virus which replicates in the upper respiratory tract as opposed to the lower respiratory tract, and replicates enough there to generate a nice immune response such that for the rest of the time you're in pretty good shape. And actually, no, I do remember what these are. The 2 wild type are going to be what? Right, HA and NA. So these are going to be whatever's circulating at this time, hemagglutinin and neuraminidase. Why? Because they're on the outside of the virion. What does your immune system recognize? What's on the outside of the virion? So everything but what's on the outside of the virion is this cold-adapted version, and what's on the outside is going to be your HA and NA. So we now know exactly what the genotype is. This was originally made, just like all attenuated vaccines, do a whole bunch of passages, see that it doesn't give you much disease, use it. But more recently, we found out where those mutations are, and curiously enough, they're mostly in these polymerase genes. So polymerase genes are determining whether you replicate at the low temperature or replicating at the high temperature, which is kind of the last thing I would expect in terms of how you'd get these to, in fact, work. So these are the various genes and the changes that you have there. I won't talk about pandemic flu, even though it's frighteningly interesting and important and all that good stuff, but that's basically because it's much less of a problem than just annual flu that we have to deal with anyway. So with that, have a nice rest of the day, and we'll see you on Wednesday.